Welcome dear students. I, Dr. Shah, Ubaidullah, Assistant Professor Biotechnology at Government Degree College, Pulwama, welcome you all to this lecture on transcription. Here we will give you an overview of the process of transcription. So, you must be knowing that transcription, it is the synthesis of RNA from DNA, and this is one of the important steps in the information flow in a cell. So, our objectives will be, we will be learning about what is transcription first, then we will talk about the comparison between the process of replication and transcription, what are the differences between the two processes and what are the similarities between transcription and replication. We will talk about, since you know that the DNA has two strands, we will talk about the coding strand and the non-coding strand, and then we will also talk about the enzymes which are involved in the process of transcription, and we will compare the process of transcription between prokaryotes and the eukaryotes, and these will be the objectives of this lecture. So, first if we talk about transcription, what is the definition of transcription? It is simply the synthesis of RNA chain from DNA molecule. So, you see here we have got DNA, and this DNA, from this DNA we have the synthesis of RNA molecule. Here it is written mRNA, but it can be tRNA, it can be rRNA, it can be mRNA. So, this process is known as transcription, and this is the primary step or the first step in the information flow in a cell, and when the information flows from DNA to protein, and this information flow, this is known as the central dogma of molecular biology. Here DNA, it gives rise to RNA, and then message in the RNA, it may be translated into the protein, or RNA may be used as such. DNA also forms a copy of itself. This is known as replication, or as you know, in some viruses we have RNA as the genetic molecule which stores genetic information. It can give rise to DNA, what is known as reverse transcription, and then the formation or the synthesis of protein from RNA is known as translation. And this step, that is from DNA to RNA, this is the primary step, this is the process what we are going to talk about. That is the process of transcription or the synthesis of RNA molecule from DNA. So, first let us compare DNA replication versus DNA transcription. As you know, DNA replication, it is the process by which DNA makes copy of itself, and in transcription what happens, this message in the DNA, it is copied into RNA molecule. So, if we talk about the similarities, in both processes a new oligonucleotide strand is synthesized. So, and this new synthesized strand, it is complementary to the template strand. So, in both DNA replication and transcription, a new oligonucleotide strand is synthesized, which is complementary to the template strand. But there are also important differences between the two processes, which we will see. First, if we talk about this new strand, in case of DNA replication, the new strand synthesized is deoxyribonucleotide strand. That is, it contains DNTPs, deoxyribonucleotides. While as when we have transcription, that is RNA is formed from DNA, and this new strand, it is composed of ribonucleotides. So, this is the first difference between transcription and replication. In replication, we have the new strand which is made up of deoxyribonucleotides, while as in RNA synthesis, the new strand is made up of ribonucleotides. Then if we talk about the template strand, in the process of replication, where the DNA forms copy of itself, both strands serve as template for the synthesis of new strands. Since in replication we have to form two new strands, so both the parental strands, they serve as the template and they direct the formation of new strands. So both the strands of DNA, they serve as the templates for replication. While as in transcription, we have the synthesis of single-stranded RNA from double-stranded DNA. And here, only one of the strands serves as the template strand. And this template strand, it is known as the coding strand. Then if we talk about the enzymes which are needed in replication, or if we talk about the enzymes which are needed in replication, the DNA polymerases which are needed in replication, they need a primer. And this primer, it provides free 3' OH group to which incoming nucleotides are attached. But in case of RNA synthesis, that is transcription, the enzymes, they do not need a primer. Or in other words, I can say RNA polymerases or the enzymes which are involved in the synthesis of RNA, they are capable of de novo synthesis. De novo synthesis means that we do not need any primer to which the incoming nucleotide is attached. RNA polymerases, they are capable of de novo synthesis of RNA. And then if we come to these differences, transcription copies only a certain portion of a genome. That is, if we have got this gene may be copied and RNA may be formed. And it is not necessary that whole of the genome will be copied, as it happens in replication. In replication, what we have, we essentially form another copy of the genome. So whole of the genome is copied. But it is not true with transcription. Here only a gene or a part of the genome is transcribed. So this is the process of transcription. We have got DNA, double-stranded DNA, and then what happens? There is unwinding, and then one of the strand, it behaves as the template, and this red one, it is the sequence of the RNA which is transcribed from this DNA. And if we see this template, it behaved, this strand, it behaved as a template, and this strand, it is known as the coding strand. So only one strand behaves as a template for the synthesis of RNA. While as in replication, as we have discussed, both the strands behave as templates since we have to form both the strands. But it is not the case with the in transcription. Then let us elaborate about the coding and the non-coding strands. So if we talk about the coding strand, the DNA strand whose base sequence is identical. Identical does not mean same, since we do not have T in RNA, it is replaced by uracil in RNA. So coding strand, the DNA strand whose base sequence is identical to the base sequence of the RNA transcript produced. So if we talk about the base, this coding strand, its polarity is 5' to 3' direction, and this is also known as the positive strand, the sense strand, or the non-template strand. So all these are the names for the coding strand. So it can be positive strand, it is known as templates, it is known as non-template strand, or it is known as sense strand. So here we can see we have a non-coding strand and the coding strand. If we talk about the non-coding strand, its direction is 5' to 3'. If we talk about the coding strand, its direction is from 3' to 5', since RNA will be synthesized in 5' to 3' direction. So if we compare the sequence of the RNA, we will see it is identical. Only the difference will be that thymine will be replaced by uracil, and its sequence will be same or identical as that of the non-coding strand. Then if we talk about the non-coding strand, it is the strand whose base sequence is complementary to the RNA transcript produced. Its direction will be in 3' to 5' direction. There are another names for this strand. This is known as the negative strand. This is known as the antisense strand, or this is also known as the template strand. Since it is this strand which is in 3' to 5' direction, which acts as a template for the synthesis of RNA, which is synthesized in 5' to 3' direction. So here we have shown the two strands, 5' to 3', it is referred as plus strand. It is also known as coding strand. It is also called as sense strand, and it is the non-template strand. While as the strand which is in 3' to 5' direction, this is known as template strand, since it behaves as a template for the synthesis of mRNA. It is known as antisense strand. It is known as non-coding strand. It is known as minus strand. Then if we talk about the enzymes, transcription enzymes. So enzymes which catalyze the formation of RNA, they are known as RNA polymerases, or we simply call them as RNA pol, and we write them as RNA-P. RNA pols and perform the same function in every cell. That is, their main role is to catalyze the synthesis of RNA from and RNA pols, if we talk about from bacteria to humans, they are multi-subunit enzymes, exception being some phages which have a single subunit enzymes. First, we will talk about the bacterial RNA polymerases. Bacteria being simple, prokaryotes, they have got a single RNA polymerase, and this single RNA polymerase, it transcribes all the genes or all the types of RNA, tRNA, mRNA, or rRNA, or any other type of RNA, it is all synthesized by this single RNA polymerase found in the bacteria. If we talk about the core enzyme, it consists of 2 copies of alpha subunit. That means we have got 2 alpha and a copy each of beta and beta prime. So we have 1 copy each of beta and beta prime, and we have 1 more omega subunit. So this is the composition of the core enzyme. If we talk about the complete enzyme or the holo enzyme, it contains 1 more additional subunit, that is the sigma factor. So if we talk about the composition of the holo enzyme, it has got 6 subunits, while as if we talk about the core enzyme, it has got only 5 subunits. So here we have shown the subunits of E. coli RNA polymerase. So 2 alpha, 1 beta prime, 1 omega, and together with the sigma factor, they form the whole enzyme. So if we talk about in detail bacterial RNA polymerase, so the 2 alpha subunits, they help in enzyme assembly, interactions with the DNA, and can bind some activators. Activators are some proteins which may influence the rate of transcription. So if we talk about the catalytic center of the enzyme, that is RNA pol, it is formed by beta and beta prime subunits. So they have the important role in catalysis. Now if we talk about the sigma subunit, it gives promoter specificity. That means it decides which gene is to be transcribed or which promoter has to be used. So holo enzyme initiates transcription from promoters only, while as the core enzyme, which is having 5 subunits, it can initiate transcription from any point. So this is the sigma factor, core enzyme. With the sigma factor, it forms the holo enzyme. So if we talk about the sigma factor of bacterial RNA polymerase, so in bacteria, there are different sigma factors which are used. So the predominant sigma factor is known as sigma 70, and this 70, it refers to the molecular mass of this subunit. Since its molecular mass is 70 kilodaltons, so that is why this subunit is known as sigma 70, and it is most predominant or most common sigma factor in bacterial cells. There are other sigma factors which are expressed under different conditions, and different sigma factors, they bind to different promoters, hence different genes are transcribed. If we talk about one such sigma factor, that is sigma 32. This is expressed when the bacteria is under heat shock. And then we have one more sigma factor, that is sigma 54. If a cell is nitrogen starved, then we have got sigma 54 which is used. And then we have one more, that is sigma 28. It is used for the expression of the flagellar genes, or the genes which are involved in the synthesis of flagella. So we can conclude that use of alternate sigma factors like sigma 70, sigma 32 and the sigma 54, they have a role in controlling gene expression. They decide which promoter has to be used and which gene has to be expressed. Then if we talk in contrast about the eukaryotic RNA polymerases. So if we talk about eukaryotic cells, they have got at least 3 different RNA polymerases. And these different RNA polymerases, they synthesize different types of RNA. And these RNA pols, they are named as RNA pol 1, RNA pol 2 and RNA pol 3. In addition, plants have got 2 more, that is RNA pol 4 and RNA pol 5. And all these 5, they are multi-subunit enzymes like the bacterial counterparts. If we talk about eukaryotic RNA polymerase 1, this is located in the nucleolus, and this is involved in the transcription of 28S, 18S, 5.8S rRNA genes. So this mainly transcribes the ribosomal RNAs, and 28S, 18S and 5.8S are transcribed by RNA pol 1. And if we talk about the sensitivity to alpha amanitin, RNA pol 1 is insensitive to alpha amanitin, which is a poison. So this RNA pol is not affected by alpha amanitin. Coming to eukaryotic RNA pol 2, this is located in the nucleoplasm and it transcribes protein coding genes, that is mRNA. It also transcribes genes for small nucleolar RNAs which are known as snRNAs, except U6. U6 is not synthesized by RNA pol 2. It also is involved in the synthesis of small nuclear RNAs, SNO-RNAs and microRNAs. And this enzyme, it is highly sensitive to alpha amanitin. Even a small concentration of the order of 1 microgram per ml is enough to inhibit this enzyme. Coming to RNA pol 3, this is located in the nucleoplasm and it transcribes genes for tRNA, 5S RNA, U6 snRNA and 7S RNA, which is associated with SRP, that is signal recognition particle. And this is moderately sensitive to alpha amanitin. This is inhibited at a concentration of 10 micrograms per ml. If we talk about another 2 polymerases, that is RNA pol 4 and 5, which are found in the plants, they are enzymes that synthesize small interfering RNA, that is siRNA, which is involved in the gene silencing. If we talk about RNA pol 5, it is also plant specific RNA pol found in the nucleus, and they together with RNA pol 4, they are required for normal function and biogenesis of small interfering RNA. Pol 4 is involved in siRNA directed DNA methylation pathways which lead to heterochromatic silences, that is they are involved in regulating gene expression in plants and gene silencing. Now at the end of this lecture, let us compare bacterial DNA RNA pols versus eukaryotic RNA polymerases. So if we talk about bacterial RNA pol, which we discussed, it is a single enzyme and it transcribes all types of RNA, be it mRNA, tRNA or rRNA. So small enzyme, single enzyme in bacteria, it transcribes all types of RNA. While as in eukaryotes, we have got at least 3 different types of RNA polymerases and each synthesize a different type of RNA. If we talk about the core enzyme in bacteria, it has got 5 subunits. While as in eukaryotes, all the enzymes, they are multi-subunit having more than 10 and up to 20 subunits. If we talk about the initiation factor, in bacterial RNA polymerases, we need only one initiation factor that we called as the sigma factor. While as in eukaryotes, we need multiple initiation factors and together they are known as GTFs, which stands for general transcription factors. Then again, if we talk about the product which is being synthesized, that is RNA. In case of bacteria, the RNA synthesized is polycistronic. That is, it contains more than one gene. While as in case of eukaryotes, we have only monocistronic RNA which is being formed. That is, it contains information for only one gene. Thank you. This is